Namaskar. Welcome to our channel Bio by Gulab Sir by Wisdom Coaching Center. As a part of series of lectures, today's topic is replication of DNA. Formation of two double DNA double helix. It is the process of formation of two DNA double helix from single helix. Replication of DNA. So it is a process of formation of two double helix from a single helix. It occurs during the synthesis phase of cell cycle. To understand it, as we have already studied cell cycle, here as you know that this is the 0 phase called question phase, this is the M phase, this is G1 phase, this is G2 phase, and this is called S phase, during which that replication of DNA occur, single stranded, this double stranded single DNA, form two copies. Suppose single DNA become double. As you know that monovalent chromosomes, during G1 phase, these chromosomes are monovalent. During synthesis phase, by the end of synthesis phase, same chromosome become bivalent. It means that DNA material within that chromosome become double and form a sister chromatid. This formation of sister chromatid, the two sister chromatid in bivalent chromosome are produced from each other during replication of DNA. In another way to understand, this is a parental DNA. It get bifurcate, and that two parental strand get separated from in the process, and that I am going to explain. And two new DNA strand, that two new DNA helix are produced. These are the two new helix which are produced. So two parental and two new one, that means 50% parental, 50% new, that few new are produced from new nucleotide which are added in the sequence according to the process which I am going to explain. So once again, replication of DNA occur during synthesis phase of cell cycle, where that one DNA double helix become two DNA double helix, where monovalent chromosome become bivalent. To understand this process, first of all, let you know that replication of DNA, that entire length of the DNA, just imagine we have 6.6 into 10 raised power 9 base pair. 6.6, how many total base pair we have? 6.6 into 10 raised to the power 9 base pair occur in 8 minutes. It is given, that is 2000 base pairs join in a DNA fragment per second, such a speed. So three things are very important during the replication of DNA. One is that speed, such a speed, very high speed, that I did explain, that entire genome human with diploid condition, that 46 chromosome, 6.6 into 10 raised power 9 base pair get replicated within 8 minutes, that is 2000 per second speed. Second, accuracy, because the DNA which not only going to replicate but also duplicate the information present on that gene to represent each cell after cell division, when one cell form two cell. During this cell cycle, this single cell produces, after the mitosis, produces two cells. So two cells are being produced from single cell. That production of these two cells with same genetic makeup, that required the accuracy. As we are in zygote stage, undergo division and redivision, we grow and develop, become the fetus, we take the birth and become the adult by the process of this cell division, with the 100% accuracy of that replication of DNA to represent in each cell, newly produce each cell. So accuracy. Third point is that high level of, or very high energy requirement. It required tremendous energy and that supplement energy is supplied through ATP molecules. So adenosine triphosphate is not only act as a energy molecule but also act as a nucleotide, one of the nucleotide in a DNA, and that we are going to understand. Now see once again, replication of DNA is the formation of two DNA double helix structure from one single DNA. It occurs during the synthesis phase of the cell cycle, and here that I have shown that how one DNA become two DNA, and the speed of the DNA is 6.6 into 10 to the power 9 base pair get replicated within 8 minutes in our body, that is 2000 base pair per second, such a speed with accuracy and requirement of energy. Entire DNA cannot be bifurcated. Suppose, to understand, I draw here this chromosome, and suppose this has this DNA, long DNA, and to bifurcate that DNA to generate the new strand is not possible for entire length. This is suppose a bifurcation for entire length at a glance, it is not possible. Therefore, by forming replication fork, DNA of replication begins. Origin of replication has specific position in plasmid and circular DNA in bacteria. Same way, DNA of replication is not decided by on that DNA, but it is the condition present inside the cell. As well as the position of replication is fixed. These 2 points also you have to remember. Now to understand replication of DNA, suppose this is a DNA. I am not drawing in a helical form, I am drawing simple in a straight, and this is a replication fork which produced by the part of that process. That process includes breaking of hydrogen bond between complementary base pair, uncoiling and unwinding of DNA structure, joining of primer in presence of primase, formation of continuous strand and formation of discontinuous strand, that I am going to explain as a step. To mention point, in a point that first is breaking of hydrogen bond, breaking of hydrogen bond between complementary base pair. Suppose here there was a adenine, naturally here there would have been a thymine, here guanine, here there would have been cytosine, cytosine, here guanine, here guanine, then cytosine, here thymine, then here it would have been adenine, here thymine again, here adenine, here adenine, here thymine. So see, these were the complementary base pair, and they were joined by double hydrogen bond between thymine and adenine and triple bond between guanine and cytosine. So that bonds are breaking of hydrogen bond by helicase enzyme. Which enzyme? Helicase enzyme is very important. Then unwinding of DNA. Second step is unwinding of DNA. Unwinding of DNA double helix. DNA double helix. It is caused by or catalyzed by specific enzyme called topoisomerase. Topoisomerase cause unwinding of DNA. So breaking of hydrogen bond between adenine thymine, guanine cytosine as a complementary base pair or complementary nucleotide with that complementary base pair is by helicase, and now DNA which was double helix in a helical form, it unwound, and now this strand gets separated to generate the new strand on them, and these strands are in this opposite orientation 3-5- and 5-3-, 3-5-, 5-3-. So third step is attachment of primer or initiation. Attachment of primer, also called as initiation, in presence of enzyme primase. In presence of enzyme primase. So what is primer? Is a small stretch of, it is a small stretch of nucleotide with complementary to the nucleotide on template strand. So this is a template strand complementary to it thymine, cytosine, guanine. So few nucleotide, 10 to 15 nucleotide in a chain, in a small fragment is called primer. So this is a primer. This is a primer. It join in presence of enzyme primase. Fourth stage is formation of continuous strand. So fourth stage is formation of formation of continuous strand, also known as leading strand, also known as leading strand. What is that leading strand? It is catalyzed by DNA dependent DNA polymerase enzyme. DNA dependent DNA polymerase enzyme. This is a highly efficient enzyme. This enzyme is highly efficient enzyme because it has to carry out or catalyze long process, because thousands and thousands of lakhs of nucleotide to be joined complementary to the template strand continuously till the end of that DNA, and DNA length may be with millions of base pair. So DNA polymerase has high efficiency to have the complete catalysis, complete control over the system, to have continuous formation of continuous strand on this template strand till the end of the DNA. So therefore DNA polymerase is said to be highly efficient enzyme. When this continuous strand formation of this continuous strand is continued with the addition of nucleotides complementary to the nucleotide present on that strand, template strand, by the time Okazaki fragments keep on forming complementary base pairing with the, here suppose this adenine, here will be adenine, here thymine, here guanine, cytosine, guanine. Now these are the complementary nitrogenous bases and nucleotide which join with this nitrogenous bases present on another strand of DNA in 3' 5', but overall formation of new strand, as here this is 5' to 3', here also the new strand is form 5' 3'. So overall direction of synthesis of new strand is 5' 3', either in leading strand or lagging strand. This is fifth state, that is what formation of discontinuous strand. This is so called discontinuous strand, so called it is because of this Okazaki fragments. What are these? These are the Okazaki fragments. They join to the nitrogenous bases present on another strand in 5' 3' orientation. But later these Okazaki fragment join in presence of enzyme ligase. Therefore here I am going to write enzyme ligase that catalyze the process of joining of Okazaki fragment on another strand. Thus these two strands on two parental strand, these are the parental strand and these are the new strand. So these new strand which are produced on this parental strand, this is this one and this one, these are the new strand which produce on this parental strand, is continued till the end of the DNA fragment, catalyzed by DNA dependent polymerase, as well as that Okazaki fragments are join each other by forming phosphodiester bond in presence of a ligase. It's continue on strand, another strand. Thus formation of leading and lagging strand is part of the process which continue till the end of the replication of complete DNA to form the two DNA double helix structure from single DNA. So this was a single, now going to produce the two. This was a single, produce two. This was a single, produce two. It occurred during synthesis phase. Now here, role of ATP molecule, very important to understand. Role of adenosine triphosphate is not only provide energy, but also it act as one of the nucleotide. This is very important to understand. This is not only a energy supplement molecule, but also part of the nucleotide. As you know that there are 4 nucleotides that they become the part of DNA. Adenylate acid, that is adenosine triphosphate, guanylate acid, cytidylate acid, and thyminoic acid. So thymine nucleotide, guanine nucleotide, cytosine nucleotide, and adenine nucleotide, that they are the part of. Therefore adenosine triphosphate is a adenylic acid. It is a one of the nucleotide, not only become the part of DNA but also provide energy which required for formation of hydrogen bond between complementary base pair as well as ester bond between two subsequent nucleotide on the same DNA helix. And this is the entire process occur within 8 minutes in all the chromosome present in our body, and that take the time 8 minutes. So know it that by or recognize it with its speed, that it is with accuracy and supplement of energy occur during synthesis phase of the cell division. So every time when there is a regeneration and repairing, when there is a growth and development, when there is a development of zygote into the fetus and adult, or when the diploid organism they grow and develop, or any haploid organism when they reproduce, that they need to have the duplication of material. So every time that DNA material become double, and then only that single cell become double, two cell, or that cloning of the cell occur. So entire process of growth, development and reproduction, that they require to have the duplication of DNA material. Without that interphase, that means preparatory phase, during which protein RNA synthesize as well as DNA material, nuclear material also become double. So becoming double is very important requirement of the reproduction of cells. So replication of DNA is a part of process of not only that simply to become the material double, but also part of the growth and development. So here we did understand replication of DNA. Once again, to summarize it, that formation of two DNA double helix structure from single double helix is known as replication of DNA. It occurs during synthesis phase. It takes 8 minutes or 2000 per second. It has 5 steps. One, breaking of hydrogen bond, so as to get separate those two strands, unwinding of DNA so that it gives chance to have the attachment of primer in presence of enzyme primase, and formation of continuous strand in presence of DNA dependent DNA polymerase, as well as joining of that opposite fragment in presence of ligase enzyme, so as to have complete synthesis of DNA double helix. As I told you, that name of the enzymes also, please remember this name of the enzyme as a part of NEET. These events also you have to remember in a sequence, because question can be asked in NEET. One of the following is the correct sequence of action: formation of DNA replicate two DNA double helix and catalyzed by the group of enzymes. So one of the following is the sequence of enzyme they catalyze the process of replication in a sequence. Or question can be asked, what is the speed of DNA replication? What is the role of ATP molecule? What are the three things which are the very important part of the DNA replication? Is maintenance of speed, accuracy and energy requirement. So this process being very complicated but occur with very accuracy. If sometime it so happen that any wrong base pair is get over inside this replication, suppose in place of cytosine, if adenine is get over here, then that process which is continue, once again that come back, replace that wrong pair, base pair, by cytosine instead of adenine, and once again it is continue. That is called as repairing of DNA. So that is called correction of DNA. So this is also carried out by DNA on its own. So that is very important part of the replication of DNA. So repairing of DNA is also occur on its own. I hope you did understand entire process of replication of DNA. Even if you are in doubt, you can ask in comment section, and I appeal you to like our channel by subscribing it, and by pressing bell icon you are notified, we will be notified time to time as and when the new videos will be uploaded. Thank you.